Hi and Assalamu alaikum. My name is Nadratun Naim. Today we present for individual presentation for AQU3454 Aquaculture Biotechnology course on the topic vaccine in aquaculture industry. So, for the introduction, how does the vaccine itself develop? It is required a strong understanding in fish vaccinology. It's generally based on two main sciences, which is microbiology and immunology. With the enhancement of molecular biology and the improved knowledge of proteins, antigen, and all, there are rapid development for new generation of vaccine for use in animal and humans. Which is in this presentation, we focus on aquaculture industry. So what is important of vaccine or the advantage of vaccine? It is no environmental impact. Theoretically, it can control any bacteria or viral diseases, and there's no accumulation of toxic residues on the organism, and no toxic side effect, and its efficiency or its effect for the longer duration. And vaccine are used to prevent a specific disease outbreak from occurring, and vaccine is not a therapy, and unlike the antibiotic, generally will not be effects when to stopping a disease outbreak once it has begun. So vaccine has two types. The first one is conventional fish vaccine, which is contribute of inactive or kill vaccine and live vaccines. The second one is alternative vaccine technology or modern vaccine, which is subunit vaccine and nucleic vaccine. That is RNA and DNA vaccines. Okay, so now we will go further for inactivate or kill vaccine, which is it is typically created from a virulent disease causing microbe, and through some process it loses its ability to infect. And then it is inactivate vaccine are more stable under field condition than live vaccine, and may be less expensive to produce, and they are usually found to be safe, but may induce weaker or shorter life immunity when compared other vaccine type, because it do not persist within the environment or in the vaccinated fish. And as for their advantage, it is include the potential for immunosuppressive messenger antigens, and it is can reduce immunogenicity due to the denaturation of proteins and systemic reaction to certain adjuvant. So for the next conventional fish vaccine, which is live vaccine, it is prepared from one or more viruses or bacteria displaying attenuated virulence or natural low virulence towards the target fish species, usually using physical and chemical process. Live vaccine tend to be more immunogenic than cold preparation due to their ability to proliferate or enter the host and stimulate greater cellular responding to both innate and adaptive immunity, and it can generate a strong antibody response because it is such a cell-mediated immune response are considered to mimic a natural pathogen infection, and it is almost identical. And it is a live vaccine are usually quite effective and retain attribute of natural infection, and no adjuvant are required because the use of an accompanying adjuvant is not typically required to enhance efficacy. And live vaccine have a greater potential to be administered via oral or immersion system, especially for commercial application for aquaculture. So for the next one, we will discuss about alternative vaccine technology or modern technology that comprise of subunit and nucleic vaccine. First, we will go further for subunit vaccine, which is subunit vaccine take advantage of using only antigenic component for vaccinations. And since subunit vaccine cannot replicate in the host, there is no risk of pathogenicity to the host or non-target species. Subunit vaccine can be produced in a highly characterized state, and they target immune response towards specific microbial determinant, enable the incorporation of unnatural component, and can be freeze-dried. And the most important thing is allowing for non-refrigerated transport and storage. Also, virus-like particle (VLPs) are the component of an advanced tubuling formed from the self-assembly of viral capsid protein into particle that mimic the natural structure of virus. Due to the advantage compared to the other vaccine type, the interest in VLPs technology has increased in recent years, and the studies show that VLPs with similar size and geometry to the native virus could elicit an antibody response in fish by injection. And among the VLP vaccine example is reverse necrosis virus (NVV), which is VLP vaccines that utilize E. coli, yeast, and baculovirus, and plant or cell-free self-assembled operation were produced for VLPs. For the last vaccine type, which is a nucleic acid vaccine that consists of DNA and RNA that encoding the antigens of interest and are considered relatively simple to generate and safe to administer since they cannot revert to a pathogenic state. And this vaccine have the combine of positive attribute of both live attenuate and stable vaccine. And this nucleic acid vaccine have developed for use in aquaculture over the past 20 years, and it is a scalable, competitive, and no cold storage is needed. We'll go further for nucleic acid vaccine DNA, which is DNA vaccine is consists of an expression of plasmid that carries a specific gene that codes for a selected antigenic protein. And this development of DNA vaccine can be rapid or relatively straightforward if a protective antigen is known. And the first reported DNA vaccine that used in aquaculture was for against IHN, that it was tested in rainbow trout. And the vaccine also have been efficacious in the prevention of fish exposure to intracellular bacteria, which is Mycobacterium marinum. As for RNA vaccine, it is a safe, non-infectious, and degraded by normal cellular, and there's no potential risk of infection. And there's two major RNA-based vaccine, which is the first one is conventional, which is non-amplifying mRNA. The second one is self-amplifying mRNA. So for the vaccine delivery systems, there's a three type, which is injection, immersion, and oral delivery. And we can see the highlighted point of each delivery system here. After all of the discussion, we can conclude that an ideal fish vaccine is one that is safe for the animal and environment, economical for large-scale production, especially for aquaculture, and it is easy to administer, capable of inducing a strong immunity throughout periods, and the last one is demonstrate minimal side effect to the organism. And we can see that new and alternative fish vaccines are adapting advanced technologies often developed based on the needs in animals and or human medicine, but have shown a great promise for aquaculture. Thank you.